intro for chapter 6, nucleic acid and protein synthesis. There are 5 parts in this chapter, which is the structure of DNA and RNA, the DNA replication, genetic code, protein synthesis, and gene mutation. For structure of DNA and RNA, you have to know the structure of nucleotides and the structure of DNA. For DNA replication, you have to know the 3 requirements for DNA replication, which is the first one, DNA template, free DNA nucleotides, and the third one, enzyme, which I will explain more detail in the following question. And for genetic codes, you have to remember the gene expression and the features of genetic code. For protein synthesis, you have to know that DNA undergo transcription to form RNA, and RNA will undergo translation to form polypeptide. So let's move on to the past year question. Okay. For the first question, nucleotides are structural components of nucleic acids. Each nucleotide consists of a pentose sugar, a phosphate group, and a nitrogenous organic base. Complete table 5.1 to compare DNA nucleotide with RNA nucleotides as structural components of nucleic acids. So nucleotides are monomer of DNA and RNA, and it consists of a pentose sugar, please ignore my drawing, and a phosphate group, and a nitrogenous base. So this is just a very straightforward question. So does anyone want to answer the question for the first one, which is the pentose sugar component for DNA and RNA? Please type in the chat room. Yeah, it's deoxyribose and ribose. So the pentose sugar component for DNA is deoxyribose, while for RNA is ribose. And the second one, anyone wants to try the purine basis for DNA and RNA? Yeah, yes, Pei is correct. Yep. The purine bases for DNA and RNA are the same, which is the adenine and guanine. Yeah, they have the same purine bases. So for the next one, what is the pyrimidine basis for DNA and RNA? Yep, it is for DNA it is cytosines and thymine, and actually for RNA is cytosines with uracil, not thymine actually. Thymine is only for DNA. And just a quick recap that purine bases are two rings, and the pyrimidine basis is 1 ring. Okay, so let's move on to the next question. So here's the mark scheme. So figure 3.1 is a diagram of a monomer of the nucleic acid messenger RNA. So name D, E, and F in figure 3.1. So this is just like the previous question that I've mentioned. Here is, anyone wants to try? Please name the D, E, and F. Yes, D is here, D, E, and F. Yep. D is actually, yeah, D is, so let's see the mark scheme. D is the nitrogenous base, not ribose. Ribose is this one, which is the sugar. So E is the ribose, and F is the phosphate group. So let's move on to the next question. State one way in which the structure of DNA differs from the structure of RNA. So in this question, you have to know about the structure of DNA, which differs from messenger RNA. So here's a diagram of DNA. So for the structure of DNA, for the structure of DNA molecule, DNA is made up of two long strands of anti-parallel nucleotides in which opposite complementary bases pair up by hydrogen bond, and to form a ladder-like construction which twisted into the shape of a double helix. So this is a shape of double helix. So for the mark scheme of this question, so this is just a 1 mark question, so just pick anyone from the below option. Is 2 strain of polynucleotides, not one, because you can see here it is made up of 2 strain, and it is deoxyribose, not ribose, because now you are stating about the structure of DNA. And thymine, uracil is for RNA, and they have base pairs, or the structure is double helix. So next question. Figure 1.1 is a diagram showing the structure of a section of a DNA molecule. Okay. Draw a circle around one monomer of DNA in figure 1.1. So you have to circle a monomer of DNA, which you have to circle this one. You have to circle the phosphate group, the sugar, and the nitrogenous base. So name 2 bases forming the base pair in X. Here's the X in figure 1.1, and give reasons for your answers. So mark scheme is here. So the basis is either cytosines and guanine, because you can see from here is that for X, right? There are 3 hydrogen bonds in X, and there are 2 hydrogen bonds here, which indicates that this is adenine and thymine, and for the 3 hydrogen bond is guanine and cytosine. Yep, 3 hydrogen bonds between the base pair. So for the next question, the statement 1 to 5 describe the event that occur during DNA replication. So first, DNA polymerase form a phosphodiester bond. 2, DNA double helix forms. 3, hydrogen bonds break. And 4, hydrogen bonds form. 5, 2 strands of the double helix separate. Write the number 1 to 5 in the spaces below to show the order in which these events occur. The first one has been done for you. So the first one has been given, which is 3, the hydrogen bonds break. So anyone would like to try? Please type your answer in the chat box. For the process of DNA replication. Yeah, Shumin and Pei both correct. It's 35142. Okay. Yep. So the hydrogen bonds break, and then the two strands of double helix separate. Then the hydrogen bonds will form, and the DNA polymerase form a phosphodiester bond, and finally a double helix of DNA is formed. So I will explain this question more detail in the next few questions. When
We mean which year past year is that?
this is 2019.
June 19, right?
Yeah.
I'm quite curious with the answer given there because according to the latest textbook, it says that it is the DNA ligase. Point number one in the textbook says it's DNA ligase that forms phosphodiester bond, whereas DNA polymerase is the one that bring in the correct nucleotide to form complementary base pairing. So I actually wonder why they put number one here. It could be in the new textbook, they have changed this content after doing research and discovered that this statement is not correct. So just want to bring attention to all of you that in the new textbook actually indicated that it is the DNA ligase that form a phosphodiester bond and not DNA polymerase. Okay, DNA polymerase only bring in the correct complementary nucleotide to form the hydrogen bond with the complementary bases at the template strand. Okay, thank you.
Okay, thank you, teacher. So next question. When a sample of DNA was chemically analyzed, it was found that 36% of the bases are adenine, so 36% are adenine. What are the percentage of other bases? Explain your answer. Here's typing error. Answers. So besides adenine, we know that there is thymine, guanine, and also cytosine. So the question already stated that there is a 36% of adenine. So what is the percentage for the other bases? Would anyone want to try? This is actually quite a straightforward question, which is similar to Miss Hall's slides before. Yep. Yeah. The thymine. Yep. Let's see the mark scheme. Yep. So the complementary base of adenine, right? Adenine is thymine. So it must be also 36%. Thus, the other two bases must add up to 28%. Since they are complementary, so guanine must be 14%. So cytosine also must be 14%. Yes. So the next question. Yep. So read through the following account of DNA replication and then complete it by filling the spaces with the most appropriate word or words. So this question is also about DNA replication. During DNA replication, the enzyme what binds to the DNA double what? So I'll fill in this question, the answer for this sentence first. During DNA replication, the enzyme DNA polymerase binds to the DNA double helix, which is the structure of DNA. So this makes the DNA's what and breaks the what bond between the nucleotides. So anyone want to try the third and the fourth? Yes, the answer is this makes DNA unwind or you can say unzip, and breaks the hydrogen bond. So between the nucleotides. So I would like to mention here that just a quick recap, why the hydrogen bonds is formed between the nucleotides is that hydrogen bond is a weak bond and it can be easily broken to expose the bases of both polynucleotide chains for DNA replication or transcription. Yep. The next one. These nucleotides are bound together at the nitrogenous base. The base adenine binds with what and what binds with guanine. So I will just fill in this question since it's similar to the questions previously. So base adenine binds with thymine and cytosine bind with guanine. So just to remember, the answer is not uracil because this is about a DNA replication, and cytosine binds with guanine for DNA. And free nucleotides found in where bind with the exposed bases, producing 2 strings of DNA. Anyone want to try? Yep, Joyce is nucleus. We can say the nucleus shape is found in nucleus bind with the exposed bases, producing 2 strands of DNA. So the next sentence, the process is said to be what? Because in both 2 DNA strands produced, one sequence of nucleotide is new, and the other is from the what? DNA. So anyone would like to answer this question? Yes, yep, it is. The process is said to be semi-conservative. Yep, it's ready. Semi-conservative. Yeah, it's parent because in both of the 2 DNA strains produced, one sequence of nucleotide is new and the other is from the parental DNA, or you can say the original DNA. Yep, so this is the mark scheme here.
Hui Min, I think the first answer is wrong. Can you ask the whole class again? What should be the correct answer?
The first one?
Yeah.
Is it the DNA polymerase?
Wrong.
Also
wrong.
Yeah.
What's the correct answer?
Helicase.
Yeah, it's helicase. So the mark scheme is wrong. So therefore, when you do any question at all, don't 100% trust the mark scheme because you know that it's wrong already, right?
Yeah.
Even if it's a pass year, also don't 100% trust the mark scheme. Sometimes it could be wrong. Yeah. Okay.
Okay. Thank you, teacher. So this is actually a helicase. Yep, mark scheme. Yes, it's wrong. So here is the diagram shows the process of DNA replication. You can see there are 2 strains, which is the leading strain and the lagging strain, and the 2 strains are synthesized in the opposite direction. So here is the enzyme. Unwind the 2 helix, the double helix, and expose the bases, which is, this is the helicase. So the process of DNA replication is like, the first is the helicase, the helicase, right? The helicase will unwind and unzip the parental double helix to form two template strings, and the leading string is synthesized continuously in the 5' to 3' direction by DNA polymerase on DNA template string by adding three nucleotides with complementary bases. So hydrogen bonds are formed between the complementary bases, and phosphodiester bond is formed to join the nucleotides together. So for the lagging string, which moves in the opposite direction of the leading string, is synthesized discontinuously in the directions of 3' to 5'. Okay, so let's move on to the next question. So, outline the role of DNA polymerase in the replications of DNA. So, let's see the mark scheme. So the role of DNA polymerase is to join the nucleotide to extending polynucleotide or string. So the answer only can allow the complementary base pairing. And the role of DNA polymerase also is to form the phosphodiester bonds between nucleotides or form the sugar phosphate backbone. And it's also proofreading the new string and repair any mismatched base pair. I don't think we learned about the proofreading new string, so I think we can just pick the top three, which is this three. Yeah, for answer of this question. So next questions. Fluoxuridine is a drug used to treat cancers. Fluoxuridine inhibits DNA replications. So suggest why the inhibition of DNA replication is an effective treatment for cancers. So anyone want to try this question? It's okay, then I just show the mark scheme. So why the inhibitions of DNA replication is an effective treatment for cancer is like, because the fluoxuridine inhibits the DNA replications, so it stops the cell cycle or mitosis or cell division or cell replication. Yeah. So next one, this is also a 1 mark question. Where's this one? So fluoxuridine prevents thymine nucleotide from being produced. Explain why fluoxuridine does not affect the transcription. So this question is about transcription. It does not affect transcription. Is that thymine only present in DNA. There's no thymine in RNA. And uracil will replace the thymine in transcription. So the fluoxuridine will not affect transcription because transcription is about RNA. So for the next question. Okay. The antibiotic rifampicin was introduced as an alternative to streptomycin. Rifampicin acts as inhibiting the enzyme RNA polymerase. So RNA polymerase is the enzyme used in transcription. So this question is also about transcription. So explain what is meant by transcription. So we already know that DNA, right? DNA itself will undergo replication, and then it will so let me move to the first page. So DNA will undergo transcriptions to form the RNA, and RNA will undergo translation for polypeptide. So this question is about what is meant by transcription. So transcription is a process of making synthesis of RNA from DNA template or sequence of bases or sequence of nucleotides, or you can say from a gene. Yeah, so now I will explain briefly about the process of transcription. So you can see here's the diagram, indicate the transcription process. So there are a few steps for DNA transcription, which is the first one. The DNA will unzip at the beginning of the gene. And the second one, which is the DNA polymerase, we use DNA's template strain to add complementary free RNA nucleotides, you can see from here. By complementary base pairing, 5' to 3' direction. So the RNA will then leave DNA at the end of the gene, like here. Yep. So I think that's it, and that's all for my part for today's chapter 6 revision. Yeah.